learning here. I'm in my second month of my internship, and this is today I'm presenting my topic, which is related to fine-tuning ChemBERTa, a large-scale supervised pre-training model for molecular property prediction. Okay. So some of the key terms that we have to familiarize with before we get into the integrities of the whole slides are one is smiles. So it's going to be a little oriented towards organic chemistry because we are dealing with molecular biology, but it's going to be an interplay of both, molecular biology the intervention of AI with molecular biology to make things to make things understandable. So the first thing that we are going to familiarize ourselves is SMILES. SMILES is abbreviated for Simplified Molecular Input Line Entry System. This is a representation of chemical compounds in the form of a line notation to describe the structure of the chemical species using short ASCII strings. So, with in the upcoming slides, I will explain this in greater details using demonstration of an image to give you more clarity on this. So, for now, I'm just giving you the key terms. Second one is BERT. So BERT is defined as bidirectional encoder representation from transformer. So BERT is an architecture where it's a transformer architecture which has proved to be creating dramatic improvements in the state-of-the-art LLM models. And this was first introduced in October 2018 by researchers at Google. So this is another variation of utility of BERT that we are going to discuss today, which is called ChemBERTa. So ChemBERT is applied to chemical SMILES data for drug design, chemical modeling, and property prediction. So basically, in a ChemBERT model, it has been pre-trained to to chemical compounds and molecules that can aid for precision medicine, drug design, chemical modeling, and drug discovery. So this is a very effective large language model that can be used by a lot of the drug discovery scientists to administer the most effective drug to cure certain diseases. So that's the utility of ChemBERT. Another term that will be coming across more so often is PIC50. So PIC50 is nothing but the maximal inhibitory concentration. So it is a concentration of an inhibition where the response is reduced by half. So when you're administering a drug to a certain to cure a certain disease at target area, how much effect will that particular chemical compound in that drug how much of those concentration of that chemical compound is required to at least reduce 50% of the effects of the damage caused by the pathogen. So this is calculated by a function called PIC50, and we will look into the formula on how it is being calculated and what is the significance of this calculation in the later slides. So as of now, I'm giving you a brief overview that PIC is a half maximal inhibitory concentration, and IC is the concentration of the substance. So P stands for potency, and so potency of the drug is being calculated using this formula. And next key term to be familiarized with is tokenizer. So tokenizer is nothing but it prepares inputs for natural language processing model. So as you have all come across while using a large language model, we use different kinds of tokenizers to tokenize certain certain words or even alphabets within a word, to to gain more perspective or understanding, or making the large language model understand the character or a sentence way more efficiently. So in order to do that, there are lots of tokenizers out there that Hugging Face. And one such tokenizer that we will be coming across for ChemBERTa in today's meeting would be byte pair encoder. So this is a default tokenizer in ChemBERTa, and it's a hybrid character and word level representation. So which means that, chemical compounds are basically carbon hydrogen atoms put together, maybe in like strings. In when an LLM model is able to interpret a chemical compound, it is able to see it as strings of characters, and a minute change in certain character can change the whole configuration of a certain chemical compound. So in order to correctly tokenize a certain chemical compound, they have created an tokenizer called byte pair encoder, which is basically motivated by the intuition that unknown words can often be decomposed into multiple subwords, and the best segmentation by iteratively and greedily merging frequent pairs of characters. So, yeah, now I'm coming back to how we calculate the PIC value. So PIC, as I mentioned in the previous slides, PIC stands for potency of a certain drug at 50% effectiveness. Sorry for the noise there. So PIC is calculated as negative log of the IC50. IC50 is the concentration of a certain chemical compound uh, acting at 50% of the area of the target binding area. So IC50 measurement tells us about the concentration at which a drug is able to inhibit a particular biological process by 50%. So say you're administering paracetamol for migraine. The chemical compounds that are present in paracetamol, all of these paracetamol, I mean, all of these chemical compounds, the IC50 is basically the concentration that is required of all of these key ingredients that are present in paracetamol that can reduce the effectiveness of that target bind by 50%. So that is what is IC50. Now, PIC and IC are negatively correlated because there's a negative log, natural log in between, right? So, um, so it is inversely proportional. So what we can understand from this is that the less the IC concentration, the more the PIC value, and the better the effectiveness of the drug. In later slides, I'll be showing you a CSV file where I have the smiles of the chemical compound along with the PIC values, within which you will be able to understand how PICs, make a significant amount of uh, uh, like how chemists or biologists or even AI engineers of that, as a matter of fact, can interpret PIC values to effectively administer a certain drug to cure a certain disease. Now I'm coming to smiles. So smiles, as I explained in the previous slide, smiles is abbreviated for simplified molecular input line entry system. So basically, in this example, you can see as this diagram, you can see this diagram here. And this is the diagram for a chemical compound called ciprofloxacin. So you can see in the A figure that you have an so in organic chemistry, when you have a I'm pretty sure everyone has come across benzene rings, but this has nitrogen in element in it. So this is an azine ring. So I'm giving just a quick trivia. So this is an azine ring, and whatever benzene ring is attached with fluorine that is called fluorobenzene ring, and this compound is something else, which is I'm not sure, but all of these rings attached together constitute of ciprofloxacin. And so what the smile architecture does is that it decomposes this supposedly uh, complex chemical compound into smaller fragments, and eventually it creates a line system for the machine, for the large language model to understand or interpret the chemical compound. So as you can see here, the start, these numbers denote the start and the end of a certain ring. So you can see 1 1 2 2 3 1 1 2 2 3 3 and 4 4 being denoted over there, indicating the starting and the ending of a certain ring. And as you see in the C figure, the green color is the main branch of that ring. So you it is denoted by the main branch there, and then you have several sub branches which is denoted by different other colors. And uh, and in the next step you can see that it is given a line notation where characters are being utilized. And if you can see, it is all going chronologically, where it starts from N1, and then all of these atoms are CC atoms. So these are carbon atoms. So N1 CCN, and then to complete the whole benzene ring, they have put a bracket to identify this particular branch, which is the light green. I think it's a cyan color, and it is denoted as C1. So this carbon is attached to the end of the one string. So this is how, with the example of this small ring, I'm able to show you how this part of the this part of the complex chemical compound, which is ciprofloxacin, is being denoted in terms of a smile string. And yeah, so this is how machine interprets complex chemical compounds into strings of an array of strings. Now, so now we are getting into the significance of predicting binding sites on bioinformatics and drug discovery. So, why do this in the first place? Why are we getting into fine tuning of ChemBERT, which is a chemical, which is a chemical encoder transformer model? Why do we have to do this? So, the importance of doing that is it comes in multifolds, where if one needs to understand the biological process, they need to understand the different reactions that go under that happen when this comes into the to the basics of drug discovery, where it gets into the enzymatic reactions of the pathogens and the particular molecule in a certain organism that is getting interacted with the pathogen, and these are called target binds. So then there is something called cell signaling and gene expression. So all of this helps with researchers to understand the underlying mechanisms that is able to correctly predict certain binding sites. And with the advent of ChemBERT, which is a transformer model, you're able to administer the right drug by utilizing this the chemical compounds like ciprofloxacin in terms of a smile string. I hope everyone is able to comprehend, because it can get a little biological, but the beauty of this is that when AI is intervened, the utility of this whole transformer model is really great for drug discovery, which is why I'm getting into a little bit of details. Second point is drug target identification. So to identify proteins with certain functions that can lead to discovery of potential drug targets. So to we have different in a human in a human body, we have different targets in our cell body, which needs to be administered when there is some sort of pathogen attack, right? So in order to do that, we need to correctly identify the right target where the drug can be administered in more efficiently, and this helps with that. So fine tuning a ChemBERT model will definitely help in understanding where the target binding sites are, and which molecule would effectively if implemented, if utilized in a certain drug, can effectively target a certain binding area so that the disease can be alleviated. So this helps researchers. And third, this paves way towards personalized medicine. So personalized medicine is become a new fad where these days everyone's body are operating at different levels, um, and everyone has a different genetic variation and protein function. So in order to um, get into the intricacies, or to feed to better to better treat a certain patient, we need to create a personalized medicine system where it could tailor the treatment for individual patient based on their unique genetic profile. So sooner or later, with effective precision medicine techniques, we can come with unique medicines that can target certain binding sites that are affecting certain patients who are suffering from certain disease. Then, last but not the least, it is used for biotechnology and biomedical research, where research is extensive about the various applications of biomedical research that include protein engineering, biomarker discovery, and development of other diagnostic tools. Now we will get into the integration of ChemBERTa with binding site prediction. So the two methods that we can see here are feature extraction and multimodal learning. So in ChemBERTa architecture, this can generate meaningful representation, which is in the form of vector embeddings of chemical compounds. This in turn are used as input future features for these for this large language model to effectively focus on binding site prediction. Now, when it comes to binding site prediction, like I mentioned, the PI50 value that I had mentioned, PIC50 value that I mentioned previously, the value of that uh, would determine the potency of a certain drug. And the second thing is multimodal learning. So as a transform, as we keep fine tuning any model as that as a matter of fact, and not just ChemBERT, as we keep fine tuning a certain transformer model, it gets used to different environments, different protein sequences, and different structures of proteins, and it is able to better adapt to certain protein structures. So and find out the PI potency value and the chemical compound, the SMILES architecture of the chemical compound in a more effective fashion. So it can better detect the potency of a certain chemical compound. So as we are combining ChemBERTa with models, it is getting used to different target binding sites and is able to understand the chemical properties of the compounds and the structural and functional properties of proteins. So when it comes to structural and functional properties of a protein, different proteins in a nucleotide sequence, you have ATGC. In a human nucleotide sequence, we have four nucleotides, which is adenine, thymine, guanine, and cytosine. So all of these nucleotides play a different role, and our DNA is structured in a hexical format, right? So so like through fine tuning models, like I'm just giving an example of for the human DNA, but uh, through fine tuning the ChemBERT architecture, it is able to get adapted to multi-model inputs, where you have lot of data sets from different organisms and different gene data sets. It's introduced to a lot of protein sequences and chemical compounds. So this is the importance of integrating ChemBERTa with a binding site prediction. And last step is where we are coming to a fine tuning model. So why we need to fine tune is because it might, so ChemBERT is already it is a pre-trained model, and it is used to its pre-existing data. By fine tuning, it is able to adapt to various other data, and the more you fine tune, the more precise the model becomes, the more accurate its predictions become, and the more effective drug discovery practices become. So it is a chain effect where it improves the ability to predict which part of a protein a given compound might interact with. Now we'll get into the code. I have snippets of code in this slide, which I have shared the code, and after this webinar, you can use the code and try it for yourself. But for the sake of this webinar, I'm going to explain it through the slideshows. So basically, for the step one is data preparation. And for data preparation, we are installing three main library dependencies, which are transformers, datasets, and Torch. Torch relates to PyTorch. Uh, so um, so we have we need Smiles representation of the chemical compounds and protein sequences of structures, and the known binding sites or the binding affinity data, right? So when it comes to this is the part where I'm loading and pre-processing the data. So in this, you can see, as you can see here, I'm loading the Smiles Big Data CS.csv file. In later slides, I will show you to get, let me just show it to you right. Um, so um, Okay, it's fine. I'll show it in the later slides. So the CSV file contains four columns, out of which we are utilizing just two columns, which is Smiles and PIC15. Smiles contains the structure of the chemical compound in a line fashion, and PIC is the effectiveness or the potency of that certain chemical compound. Now we are converting into a data frame, for which we are using pandas library, because Hugging Face, while we are while we're training the model, it can only recognize in the format of a data frame. And then we split the data into 20% and 80%, 20% for test and 80% for training. So the next step is loading the tokenizer. So in this case, as you can see here, the nomenclature of this particular tokenizer is PubChem 10 million Smiles BPE 450K, which means that it is taken 10 million Smiles compounds from the PubChem repository, and it is using the encoder, it is using the tokenizer byte pair encoder with 450K tokens. So we are using a function called Roberta tokenizer, and we are defining a function to return the Smiles value for certain chemical compounds that are the raw chemical compounds. And then we are tokenizing the dataset and further investigating how the tokenized dataset looks like. Yeah. And then we are creating something called a data loader, which contains all these tokenized data sets in the format of Smile strings. So these are basically Smile strings. I'll show you in later slides how the CSV file looks like, so that you get a better understanding. So you're creating a function called data collator, and you're padding the tokens using this function called data collator with padding. Then we're creating a train test split where you're using the tokenized data set, which is used for training, and you are creating 8 batches. And for the test data set as well, you're creating 8 test batches. Then we are printing the number of training batches and testing batches. So data loader, basically what it does is, the functionality of data loader function here is, it um, it it batches and it shuffles and it performs parallel data loading. Whereas um, there's another function called data collator that pairs sequences to the same length within a batch, and the input features are tokenized smiles and labels. So labels are basically the potency of that particular chemical compound, which will be used for drug discovery later on. The next one is loading the ChemBERTa model and tokenizer, right? So now we are using from transformers. I think this is the repetition of using this same thing. I've loaded it here. And now we're creating a function to extract the particular features that make a certain chemical compound very distinct from other compounds, right? So when you're defining a function called extract features, where we whatever you can see here, I have used an example here as carbon carbon oxygen. So this is a chemical compound that I'm passing inside the extract features function, and able to extract the features that are underlined in this particular chemical compound, to find out what is the significance of the chemical compound and what the potency of that chemical compound would look like. So this function is to extract the features that is distinctive for a certain smiles chemical compound. And then we are creating, wait, I think, sorry. Yeah. Um, yeah. Uh, Yeah, so then we are doing the optimizations. So here is where we are using either GPU or CPU server. So since I have been using Google Colab to perform all my code, I have been using the GPU administered within Google Colab, but you can also use CPU. But I would suggest using a GPU server, because to perform computationally intensive tasks, especially certain things like which is related to chemical compounds and things like that, I would suggest you to to use GPU. So this line of code is for that. And last, this is the most integral part of this code, where we are training the model, and the number of training steps. So the number of training steps is basically is calculated as the number of epochs. So here I'm introducing an optimizer. Sorry for going back and forth, but I'm using an Adam optimizer here, which has all the model parameters and contains all the learning rate. In this case, I'm using a linear regression model, and with a learning rate of 0.005. So four zeros followed by a decimal and a 5. And the number of epochs that I'm training it for is 3. You can the more the number of epochs, the better it gets trained towards. But I just used 3 because I wanted to show in a small data set how much the effectiveness would be. And the number of training steps is calculated as number of epochs into the length of the train data loader. So length of the train data loader is whatever you've passed here. Since we have split it into 80-20, the 80% of the data set, whatever number of rows are present there, that would be the length of the train data loader. And number of epochs, as we have mentioned, is 3. Uh, in this case, whatever model I have loaded, it has around 15,000 rows. So 80% of 15,000 comes to around 12,000. So 12,000 into 3, 36,000 steps. I have trained it for 36,000 steps. So so that is what is used here while we are creating a progress bar. So for each epoch, it is being trained iteratively. And inside this function, you can see KV values. So KV values is nothing but the attention visualization model that BERT architecture utilizes. So this is where this is getting into the fine tuning aspect, where we are tweaking the parameters inside the ChemBERT model and adding more data so that it is able to correctly predict protein structures, which is given in the format of a smile string. So when you mention KV values, these are key value pairs, and these are mapped effectively with the query. So Q is the query. So to get into the mathematical side of this, I will explain what the KQV values look like by actually like introducing you to a, a GitHub code that I found, which was related to transfer learning using ChemBERTa architecture. So we'll get to that in later slides. And now this is the evaluation part where I'm calculating the accuracy. And you can even try it using my code after the end of the webinar session, where I have calculated the accuracies, and it has come to around 96%. Here I am calculating the accuracy, and these are the results that I have been getting when I'm passing a certain smile string. So you can see here I have passed a very complex smile string compound which has oxygen, carbon. So it was a random smile string that I gave, and the predicted value was minus 0.23. This predicted value that you see here is nothing but is nothing but, so I'm trying to evaluate the pre-trained ChemBERT model, right? The fine-tuned, sorry for the misnomer here, it's the fine-tuned ChemBERT model. Now I'm introducing the new compounds here, and you can see the predicted value coming to around minus 0.23, which means that this belongs to the PIC value, which I've discussed in the latest previous slides. So yeah, this value, this is coming into a negative. So when PIC value is coming to negative, it means that it is less effective to a certain to a certain target bind to be administered for treating a certain disease. So we cannot be using that for chemical compound in a certain drug. So with this information, you're able to understand that this particular compound is not to be utilized for that certain drug. And when you're passing a smile string like CCH, it is giving a predicted value of 0.32, which means that it is fairly recognizable. The potency of that compound is fairly recognizable, although more the bigger the PIC would range up to like even 5. So with respect to that, this is even meager. So we will have to the more chemical compounds you are able to use as in terms of a smile string, you're able to predict accurately what I mean, you're able to predict the PIC value, which explains the potency of certain chemical compound. Now, the benefits of this approach. One thing is it has enhanced prediction accuracy. So when you're fine tuning a certain when you're especially fine tuning a ChemBERTa model, it is getting introduced to a lot of chemical and protein features, which is making it leverage information and potentially leading it to more accurate binding site prediction. So it can say someone is hurt, has a knee injury or something like that. It could look into the target binding site of that particular knee injury, and it could add and ChemBERT would would give information about the chemical compounds that could be utilized in a certain drug that could potentially act on that target bind in order to effectively elevate the effects of that particular disease. Second thing is scalability. Now, by fine tuning, you are not only you're not only fine tuning the pre-existing model that ChemBERT has been introduced has been used to, but you're also scaling it to a level where it is being introduced to to number of chemical compounds, and it has become a storehouse or a repository where the more it has been introduced to more diverse data sets, where it improves the robustness and generalizability of the predictions. So the more you scale, the more you fine tune, the more generalized and the more accurate the predictions can become. So there's a trade-off between accuracy of the predictions and the robustness. So that we'll get into later stages, where we can assess the number of epochs. By increasing the number of epochs, it can lead to overfitting, but we can prevent overfitting by introducing it to various other data sets, and for the same number of epochs. So the more data sets it is being introduced to, the better the model gets fine-tuned, and the better the chemical compound that is effectively used for drug discovery. Then third thing is versatility, where it can be adapted to various types of binding site predictions. So I gave an example of a knee injury. It can also be adapted to like something like brain aneurysm, or something related to neural diseases. So the more as the model gets trained, it becomes and introduced to more data sets, it becomes more versatile. Yeah, now these are the references. I will explain most of these after the slides. We will get into the ArchiveX paper. We will get into the model weight mates that are used for ChemBERTa architecture, and we will also talk about the transfer learning Google code that will give you a better idea. You can visualize it in a better way and understand what is going on the ChemBERTa transformer. So basic summary of this is that these it has been like so I it's com it's covering the complete process of fine tuning ChemBERTa on a data set with smile strings and PIC values. Smile strings, as we have mentioned, are the line entry systems of a certain molecule, which is being simplified, a complex molecule that has been simplified, and the PIC value is the potency of that particular chemical compound. So steps that we have undertaken in this webinar is loading and pre-processing the data, tokenizing the data set using a pre-trained. So we have used a BPE tokenizer, which I have mentioned previously, and we are using a pre-trained tokenizer called RoBERTa tokenizer, and we're creating data loaders, initializing the model with optimizers, and then, as we have seen in the previous slides, I've shown the training loop. And then we are evaluating the model using appropriate regression metrics, where we're introducing, in the sixth step is where we're introducing it to new existing chemical compounds, and it is able to give out the potency of that chemical compound. So yes, so that is all for the slides. How do I end the slideshow? Yeah, okay. So now I will be showing the Hugging Face, the ArchiveX paper. But before that, I'll show you the transfer learning with ChemBERT transformer. So yeah, this is a code that I found on GitHub, and it talks about it, and it shows it discusses about the effects of um, you know uh, like the ChemBERT architecture and how transfer learning. So fine tuning is a form of transfer learning. Transfer learning is nothing but so I'm giving you an example in layman terms so that you're able to understand. Say you know how to ride a bicycle, and you want to learn how to ride a motorcycle. So whatever skills that you have to have utilized to ride a bicycle, you're transferring it to the motorcycle and you're implementing it. So that is transfer learning. Now fine tuning is when you know that your knowledge about riding a bicycle is limited, and the motorcycle is even more advanced because it has gear, it has clutch, and it has all of these intricacies. So you're updating your data set in such a way that you're able to better ride the motorcycle, and you're able to adapt to the new environment. So by giving this example, uh, even ChemBERT goes through this similar fashion, where it has been pre-trained to a certain data set, and through fine tuning it is introduced to even more chemical compounds, which is through which it is able to better identify a certain chemical compound and give out the potency, which can be utilized to elevate a certain disease or act on a certain target by. So as you can see here, you can see a smile mask, right? So this is like, so you can see a mask here, and so by calling a fill mask function, they have defined a fill mask function here. So they've used this architecture. They're using the BP architecture as well, and they're calling a fill mask function, and utilizing the smiles string when you are passing. uh, so you're asking the model to predict what are the 5 compounds that can come in between this mask. So when you see this mask to a chemical compound that looks like this, and it is more probable for it to identify this masked smile compound as this compound, as opposed to the rest of the other compounds. So this mask could be a hashtag, this mask could be 1 2, or even like an equal to bracket. So these are the top 5. So you can see this token here, that's the token number. So from this you can understand that each of these smile string is given a certain token, and what identifies or distinguishes this token is this token string, where you have a hashtag, and like these are this is what makes the chemical compound unique, and this is the reason why we're using a tokenizer, so that it can better identify even the minute intricacies in a chemical compound. So that's the beauty of having a tokenizer in this case. And that is why we're having a BPE tokenizer here, which looks at each and every byte, which is why it's called byte pair encoder. And as it goes along, so this is I don't want to get into this as much, because this is about drawing the chemical compound. So whatever you see here, you can see a sequence here, and you can see a mass tokenizer here. This could be anything, but what it's able to identify this compound better as is this particular chemical compound, and it is drawing this particular compound. So all of these top 5 chemical compounds have been uh, drawn here, and the red marks that you can see here are the potential binding sites of these points are the points which is very susceptible for the pathogen to attack. So say a molecule in your body, a molecule in a body is highly reactive to a certain pathogen or a disease. We can get even more even more minute about it by looking at the particular target bind. So whatever red circles that you see here are the target sites. So this is what creates that target sites. And yeah, so you can see the top 5 models here and the specific target binding sites. So yeah, which is why it comes to this. So as I had mentioned in these slides, right, I had mentioned about um, what is that? Um, I had mentioned about a KQV value here. Yeah, so you can see a KV value here, which is used during the training phase of the model while fine tuning, right? So this is where it comes into play. This is the mathematical side of that KV value, where say, so it is basically attention. So the higher attention has to be given to the certain binding side. So theoretically, like I think I'm pretty sure most of you from the previous seminars, you've been introduced to the KQV values. But this is what happens theoretically, where when a sentence like "she's eating a green apple" is given, higher attention is given between to the part of the sentence that says "eating a green apple," and even more higher attention is given towards green apple, and because that's what distinguishes the sentence from other sentence. So when you're giving a query and when you want it highly specific in terms of ChemBERT, it would be in terms of a chemical compound. And when you're giving a chemical compound as a query, the smile strings that makes these compounds specific or are taken as the key value vectors here. So whatever distinguishes this certain chemical compound from this is a certain C here, right? So that is where the key value pair this is varying. And so the ChemBERT architecture, while going through fine tuning, it is going through that phase where you are creating a softmax function across the query, the key, and you're dividing it by the root of the decay. So decay is the dimension, or the dimension of the key value. So in our case, the dimension, giving this example, the dimension of, say we are taking 5 compounds, the dimension would be 5. So whatever key value pair that we get, we are dividing it by the root of 5, and we are creating a softmax function and multiplying it with the value, which is the chemical compound, and we're creating attention. So basically attention is probability distribution between 0 and 1. And if you're, the reason why we have a scaling factor of 1 by root dk is that it is able to limit the attention given to a certain chemical compound to probability distribution. So so whatever you see here, this value that you see here, the score that you see here, it has gone through the key query key value pair, and it has been distributed in this fashion. So 97% is given to a certain chemical compound, and the rest is distributed between the other four chemical molecules. So this is where key value or the attention visualization is coming into picture in ChemBERT. And this is a certain code in for visualizing the attention mechanism in ChemBERT. And as you can see here, this is a chemical compound, and this is what happens inside the architecture. So like I said, the smile strings are being identified as sentences in the architecture. So we are going in the in the intricacies of what goes behind the neural net of ChemBERT, right? So as you can see here, you can see sentences A, sentences B, all of this, and there is a certain batch index assigned to a certain molecule. So so what you can see here is, so we have totally 5 layers as of now. The more you fine tune, the more layers you can add to ChemBERT. So in layer 0, you can see that CCC, this particular so when you see a sentence like this, um, you know uh, it is giving all the dimensions or all the character probabilities that it can take for this particular sentence, and the sentence B as well. So and then it is creating a neural net architecture where weights have been assigned to the probabilities of what would be the succeeding or the, so when you see from S, from the left hand side, you can see which would be the most succeeding chemical like or a character that would follow after this particular CCC. So the more distinct the color is, In this case, you can see that the color is most distinct across BR, which means that it has higher probability for BR to be followed, the string BR to be followed by CCC. So this is how you're able to accurately find out so what goes behind the layers is that what chemical compound would give you the highest potency level. So PIC level is being calculated. We are taking the smiles and PIC level, and we are able to find out which particular compound, if taken the best out of this iteration, you the best iteration would give you the best potent value. So whatever value that you get here um, after prediction, like say here, whatever predicted value you get here, this particular compound is going through this architecture through various iterations, and then you're getting a probability value which is nothing but the fine-tuned like PIC value that you're getting. So you can distribute it across a CSV file, having so this is a demo of how the Smiles tokenizer goes. Attention is given like to each particular compound. As you can see, as the user is going below each and every iteration, the more distinct the colors are, the uh, the better the model, the better the smile string chemical compound would be succeeding a certain certain character. So this is how, through fine tuning, we are able to. So this is like, like how you see in X-ray. This is like a model view of the neural net architecture, and through various neurons or through various neural training, it has gone through a lot of iterations to predict the right value. So the prediction value that you see over here, the back end of it has gone through a lot of iterations to through like millions of iterations to come out with a prediction value for a certain like random smile string that you give it. So the more you fine tune, currently ChemBERT is fine tuned to only five layers. The more you fine tune it, the better it is able to predict the accuracy of that model, or better predict the potency of that particular chemical compound. So this is a neuron by neuron view, and you can see how what it's able to through softmax function. Softmax function is what QK is. So for every query, there is a key matching pair, and then it is able to identify that this particular you can see here that this is the strongest link to this particular string. So this is how transfer learning comes into picture, and this is how one can fine-tune ChemBERT model on a very small molecular data set. So while I was fine-tuning this ChemBERT model, I had loaded a CSV file, right? So whatever CSV file that you see here is basically this file. So this file is smiles for all of these. So this is how a smiles for a chemical compound looks like. These are so many. This can be there cannot be even there might not be names for these chemical compounds, which is why we need such an architecture which can detect chemical compounds. And you can see PIC values. So I have fed in a certain data set like this to ChemBERT, but this is having around 16,000 rows. So I have been fine tuning this, trying to fine tune this model using 16,000 rows, and you're getting PIC values for these smiles compounds. So this PIC value is nothing but like I said, the potency of that particular compound. So the more the PIC value, the better the potency, the more effective the drug is. So you can see that this PIC value depicts that out of the 16,000 chemical compounds that you have here, the most effective or the least effective is this compound. And so this is the least utilized, the least probable. So in a drug discovery team, when someone looks through a data set like this, they're able to understand that this particular compound, if utilized in a certain drug, is going to reap less potency, or is going to be less effective. Whereas this chemical compound that you utilize here, if you utilize this particular compound, then it is able to have a more higher potency at 50% of effect, at 50% of the target binding area. So if things are getting complex, I just want to give you an example. So all of you guys have played this game called Clash of Clans, where a certain troop of army members are sent to the battlefield to fight against uh, the enemy or the counterparts castle. So what would be more effective, sending using so much resources for training 100 soldiers to attack a certain area, or in this case, let's say 50% of area, because I'm talking about PIC 50. So 50 here is 50% of that targeted area. Or would it be better to have 10 skilled soldiers who can do the same damage, or the same sort of damage at 50% of that area? So this is, so by giving you this example of a of the game Clash of Clans, I'm trying to make you understand the the value of PIC. So PIC 50 is nothing but the potency, like the effectiveness of that particular chemical compound. In this case, chemical compound is corollary to a soldier in Clash of Clans. This particular chemical compound would be the most effective if utilized uh, in a drug or in a small pill, with more concentration. So and effectiveness, another thing that one can get confused is that the more the more effectiveness of effectiveness of a drug can be related to the more concentration that you give for a certain chemical compound, the better the drug can become, or the more potent the drug can become. But that is false, because IC15 is nothing but the concentration, or the effect. I mean, PIC is the effectiveness, but IC is the concentration at 50% of the target area, right? So when it comes to that, the less amount of concentration that you give, say, this particular compound, and it is able to react, uh, uh, you know better, and it has a PIC value of higher. So the less amount of this particular compound when fed, and the more PIC value it is given, the better it is. So the what I'm trying to say is, we'll have to always, when we are this comes into drug discovery, where when we are looking into chemical compounds, the less amount, one would think that more amount of that particular chemical compound is required, or is better to administer certain target area. But that is not true. If it is able to do the damage, or is able to do the repair, sorry, with even less amount of even less amount of that particular chemical compound. So the company can this helps in cost saving for the company as well. So this leads to a trade-off between effectiveness of that chemical compound to the cost that is required to produce that chemical compound, because most of these chemical compounds are not organic compounds, these are synthesized compounds. And so the more companies will always look into cost cutting in terms of creating the more effective chemical compound which does the repair with less concentration. So all of these answers can be done. All of this leads to an the answer lies on fine tuning a model. That's the beauty of it, where the answer lies on fine tuning a certain uh, transformer model to effectively show you the results in such a way that this chemical compound is specific for a certain drug. So having said that, I am going to conclude my webinar session. And if you have any questions, please feel free to ask. I hope everyone is able to understand whatever I've talked about. Thank you so much.
Thank you, Manal. Any questions, audience? I think it was quite overwhelming considering chemical industry.
Yeah, I mean, I wanted to tell people to brace themselves before they got into this, because it could get little intensive in terms of chemical compounds and things. That's why. But I wanted people to realize how a BERT architecture can, a BERT architecture can work on something like chemical compounds and still prove to be a great resource.
Right.
Yeah. I'm seeing only, I think 10 people left. Yeah.
So its recording will be available on our YouTube channel. I've given the link in the chat, and after a few days, definitely. But other webinars are also there, and Mrinal will keep on presenting going forward. So look forward to more sessions from him. And just last 1 minute, if anybody would like to ask any question, else we'll close.
Okay, I'll just say thank you so much. A good session.
Thank you. You have any questions?
No. So I was trying to understand that what you're talking and how I can correlate it in different other fields.
Yeah. Were you able to understand the whole like the code and everything? If you have any questions related to
No,
code,
not at code level, but concept wise, I understood why it is important.
yeah. So